Aloha everyone, my name is Manuela Cortes. I'm a fourth year student at UH Hilo and I'm here to present my research on exploring micro-fragmentation size on coral growth rates. So as many of you know, coral reefs are some of the most productive ecosystems on the planet, providing critical habitat for over 25% of all marine species, despite covering less than 1% of the surface area of the ocean. However, these ecosystems are currently undergoing a variety of threats, both on a global scale, such as ocean warming and acidification, as well as on a local scale with things like overfishing, pollution, storms, eutrophications, and pathogens, and many other things. So in an effort to counteract all the negative pressures that we're putting on these reefs, restoration projects have emerged within the last couple of decades to try to protect and restore these critical ecosystems and protect the marine life that lives among them. Coral nurseries have been one of the main things that have been implemented for these restoration projects. Particularly land-based nurseries are nurseries that will provide a controlled environment for coral growth, and it will allow the corals to grow until they're ready to be outplanted back onto the reef. Now, micro-fragmentation is a technique that has been used in aquaria for many years, and it consists of fragmenting small pieces of coral, usually to about 1 centimeter squared. So it will maximize the surface area that each coral has to grow, and then once it's large enough, it'll be outplanted back onto the reef. And one of the main benefits that they have found to this technique is that with very small amounts of wild collected coral, they can outplant large areas onto the reefs that we're trying to restore and recover. A lot of research has been put into this technique to get a better understanding of what conditions optimize coral growth. And what many people have found is that size has an important influence on the growth rates. There are benefits and drawbacks to having both small coral fragments and large coral fragments. For instance, small fragments will maximize the area available for the regeneration of new polyps to grow. And studies have shown that it increases the fragment's ability to uptake nutrients. However, because they're so small, it also makes them more susceptible to environmental stressors, and they'll have less combined resources overall. In particular, I want to show this study by Scheckler et al. From 2022. You'll see in the y-axis the change in area, and in the x-axis you'll see the initial area. And what we see for both of the species of coral that they studied is that it decreases, the growth rate decreases as the initial area increases, pointing again to this idea that smaller fragments have potentially higher growth rates. On the other hand, I want to show a study by Knapp et al. In 2022, in which they studied Montipora capitata. And on the y-axis, you'll have percent net growth shown by the box plots. On the x-axis, you'll have small, medium, and large size classes. And they saw a lot of variability in their results in terms of the percent net growth. But as you see on the y-axis on the right, that shows percent survivorship. Across all of these small, medium, and large, you see that the larger fragments had a higher survivability, pointing to this idea that larger fragments will have more combined resources and making them a little less susceptible to stressors, therefore had higher survivability. What we find from all of these studies is that the results are highly variable depending on the species of coral that was used and the environmental conditions of the nursery. So it points to a need for more research to get a better understanding in how different coral species under different conditions or different fragment sizes will react to microfragmentation. For this particular study, I wanted to focus on Montipora capitata corals, and I wanted to investigate if there's a specific size of coral fragments that would best promote coral growth as well as higher survivorship rates. So, in case you don't know, Montipora capitata, or brown rice coral, is a very common species of coral found in Hawaii Island. We collected 4 different parent colonies from Onekahakaha Beach Park in Hilo, and we chose this species particularly for its morphology, as it's usually encrusting. Therefore, it provided an easy species that we could take 2D pictures of to measure that growth. These corals were then placed in the Mop Coral Nursery here in Hilo. This is a 125-gallon tank in which we conducted daily cleaning activities such as cleaning the filters, cleaning the glass and the protein skimmer, as well as bi-weekly tile scrubs in which, along with the volunteers from the mob coral nursery, we scrubbed off the algae from the tiles that would otherwise be impeding the coral growth. Each parent colony of the 4 that I previously mentioned was fragmented into 4 different sizes. Small of about 1 centimeter squared, medium of about 2 centimeters squared, large about 3 centimeters squared, and 1 control group of about 5 centimeters squared. I then monitored the growth for a 10-month period until the fragments grew large enough to touch each other, which is the objective that we had from the beginning. But at that point, as you can see in the picture on the left, once the corals are already touching each other, it's hard to differentiate between fragments. So we concluded the monitoring there and we gathered all our data. Now, at the end of the research period, I got the percent change in size of each fragment by comparing the final size with its initial size. And what we see in this graph is that percent change on the y-axis and the different size classes on the x-axis. Now, because we had such a small sample size of only 4 parent colonies, I decided to conduct bootstrapping. Bootstrapping is a statistical tool commonly used to get a better understanding of where the true average lies within a population, and as well as the variability of each of those groups. So by bootstrapping each of the size classes, we get a better idea of where that true average lies, so we can get a better statistical comparison of which one might be higher or lower than the other ones. And what we see from the results is that, well, first of all, we see that they are all statistically significantly greater than the control groups, which is essentially showing that microfragmentation does promote faster growth rates for Montipora capitata corals in these conditions. But particularly interesting, we find that medium-sized corals were the ones with the highest percent growth, indicating what we saw from the literature review, that if they're fragmented to too small sizes, they'll be more susceptible to stressors and therefore they might not grow as much. But they are higher than the larger fragments, which didn't have that maximum surface area for the new polyps to grow. Now, to get a better understanding of all the variability that I was seeing in my data, I decided to conduct another bootstrap analysis, but this time taking into account the original parent colony that each of them was fragmented from. And what we find is that we have, again, percent change on the y-axis and the size classes on the x-axis. What I found from this bootstrap analysis is that the trend was no longer consistent across all the parent colonies. So as before, we saw that medium was always greater. Once you divide it up by the different parent colonies, we no longer see that trend. Particularly if you focus on parent colony A, you'll see that medium not only had the lowest percent growth of that parent colony, it had the lowest percent growth across the whole study. So this points to the idea that genetics is playing a very important role. And it's not just about the conditions, it also has to do a lot with the genetic makeup of each parent colony. And again, the reason why we're able to know this is because all of the parent colonies and all the fragments were susceptible to the same environmental conditions. The only thing that could have been different among them is a genetic factor that we wouldn't have been able to see if we hadn't conducted this additional bootstrapping. So the key takeaways of my study is that first we find that medium-sized fragments do grow the fastest for Montipora capitata corals. However, the effect of fragmentation is heavily dependent on the parent colony that they were fragmented from, which suggests that genetic is playing an important role in microfragmentation, and it challenges the perception of the one-size-fits-all approach that has usually been taken in restoration projects. It also brings up the ever so interesting debate of nature versus nurture, where we can no longer focus only on the nurturing aspects, such as the fragment size or the environmental conditions both of the nursery or of the reefs. We also need to start taking into account the genetic aspect of all of this. This will include taking into account the genetics of the coral and the genetics of the Symbiodinium, which, as many of you know, is the photosynthetic algae that lives within the tissue of the coral that might also be playing an important role in these coral growth rates and these differences that we saw. And studies like this are calling for the implementation of taking into account this natural aspect and ensuring that there is genetic variability in our restoration projects to ensure that they will be successful in the future. I'd like to acknowledge everyone that helped me out in this study, including the people that went to the Mob Coral Nursery on a weekly or bi-weekly basis. And I'd like to open the room up to any questions. Yes?
How far apart were the coral fragments, and why was the goal for them to touch?
So the question was, how far apart were the coral fragments and why the goal was for them to touch? So that's a really good question. They were about 2 centimeters apart from each other. The goal was that they touched because that's essentially when you would start to outplant them in a normal restoration project. That's once all of these little fragments are once again the same colony, they are large enough to be able to go back onto the reef and survive the natural stressors of the environment. Yes?
You said you collected your samples from Monecajacaja. Were there any differences in your collection sites or any microhabitat situations you accounted for?
So the question is if there was any differences in the collection sites that we took the corals from. And no, that was part of the idea of collecting from Monecajacaja is because it's a tide pool, it's the area where you can find corals is very limited, so you're getting pretty much the same environments for all parent colonies. And the other part of taking from Monecajacaja is because it's a tide pool, the environmental conditions of the tide pool fluctuate a lot during the day. And therefore we were expecting the corals that we got from there to be a little more resilient to the conditions that we would then put them under in the nursery.
That brings up an interesting because if the corals are all kind of contained in this tide pool, when it comes to dispersing themselves and breeding, could there be, since we're looking at genetics, could there be a factor where certain colonies who you think might be different might have come from the same kind of group? Is there any way to make sure that that doesn't occur?
Yeah, I would answer that in 2 parts. Firstly is, you could still take this study further and actually look at the genetic composition of each coral and the symbiodinium and explore what the DNA sequences are telling us about this story. But the other part to answer your question is you would also need to take into account the way that corals reproduce. Many of them are broadcast spawners, so they'll release the larvae or the eggs into the water column, and therefore they can disperse for many, many, many miles. So we don't really know if the corals that we looked at are ones that have been bred within the tide pool or if it's just larvae that came from anywhere else.
What is the process like of harvesting the coral from the site?
Taking it from the tide pool?
Yeah.
Well, you want to ensure that it's in the water the whole time so that, it can keep breathing. It is a stressful process for a coral for sure because you're changing its environmental conditions. So we let them acclimate in the tank for about a month before we actually fragment it. But the other thing that's very important, and thank you for this question, is we chose corals that were already dislodged or had already broken apart. These are often referred to as corals of opportunity, so that you're making sure that whatever you take is corals that probably didn't stand a chance in their natural environments, and you're kind of giving them a chance within the nursery. Okay, well, if there's no more questions, thank you very much.